Good evening, everyone. I would invite Dardi ma'am to start this session.
Good evening, students.
Good evening, ma'am.
Let me share the screen. Is my screen is visible?
Yes, ma'am. It's
Yeah,
visible.
ma'am.
Okay, great. So today we'll discuss the next topic, which is the bryophytes. So in the last class, we have completed everything about the algae. And today we'll start with the next part, that is the bryophytes. Okay, so in our last chapter, we have already discussed a few things about bryophytes, like the general characteristic and all. So here we will go into a deeper. Okay, so let's start. So let's go with the definition. So bryophytes are non-vascular land plants that are considered a crucial group in the plant kingdom due to their evolutionary positions between aquatic algae and more advanced vascular plants. So basically, these are non-vascular plants. That means they lack xylem and phloem. Okay, and they are land plants, and these are very crucial group because they have a very evolutionary position between the aquatic algae, which are aquatic, and between the more advanced vascular plants. Okay, like like the next pteridophytes. They are primarily found in moist environments, so they can survive in various habitats. Bryophytes include mosses, liverworts, hornworts. So basically, they can survive in moist environments, and you will find them in harsh conditions also. So there are we will come to the classification later on. So first, let's discuss a bit the general characteristic of bryophytes. Like fresh up. So number one, non-vascular. These are the non-vascular plants. Means they don't have xylem and phloem. Okay, and they absorb water directly through their surface. Next is life cycle dominated by gametophyte. The gametophyte means the haploid stage is the dominant phase. Like in vascular plant, like in the next, I think in the last chapter also we have discussed that pteridophytes and all, where the sporophyte is the dominant stage. But in bryophyte, you will see that the dominant phase is the gametophyte, where the gametes like male gamete sperm and female gamete egg is produced. That stage is the dominant, means that stage is has longer longevity. And this is haploid. The sporophyte is dependent on the gametophyte and remains attached to it. So here, sporophytic stage means the plant stage is dependent on the gametophytic stage and it is attached to it. Means they are not independent to each other. They are dependent on each other. So this is the life cycle dominant. Next is reproduction. So they reproduce through spores, not seeds. Means here you will not find seeds. You will find seeds in where? Gymnosperms and angiosperms. Bryophyte lacks seeds. They will reproduce through spores. So sexual reproduction involves the production of gametes in specialized structure called antheridia male. Male is called antheridia and archegonia is called the female one. Okay, these are the specialized reproduction, means reproductive structure. Antheridia male, archegonia female. And then fertilization required. This is the most important point. Fertilization require water for the motile sperm to swim to the egg. Although they can survive in the land, but for fertilization purpose, they require the water. So the motile sperm, which is the male gamete, they will swim in the water and then they will reach to the egg and they will do the process of fertilization. Means their mating will take place. So it is possible only in the presence of water. Next is simple structure, means the body is like thalloid kind of. Bryophytes have a simple structure compared to vascular plants. Means their body is very much simple, means not very much complexity. More the plant will be advanced, more you will find complex structure with no true roots, stems or leaves. Means in bryophytes, you will not find true roots and true stems or true leaves. Okay, it is like root-like, stem-like structure, not exactly root. It is like, okay. So rhizoids are basically root-like structures that anchor the plant but do not absorb water. So basically, what root does? Root absorb water, but these rhizoids, they more or less absorb water, but basically they are for the for anchoring the plant to the substrate or to the land. Next is thallus or leaf-like structure. Their body may be flat, leaf-like structure thallus in liverwort, or they may have simple leaf-like structures in mosses. Means either they might you sometimes in like primitive, you will know, you can't differentiate which is stem, which is root, or which is leaf. But in some more advanced like liverworts, you can find that it is like the body is like leaf-like structure. So basically it is a thallus or leaf-like structure, the body. So basically it is very simple structure. Next is moisture dependent. Bryophytes need a moist environment for reproduction and survival due to their non-vascular nature. As they are non-vascular, there is absence of xylem and phloem. They need a moist environment for reproduction and survival. They are desiccation tolerant and can survive drying out by rehydrating when moisture returns. So basically, they can survive in the harsh environment also. Next is lack of lignin. They do not have lignified tissue, which means they do not grow tall like vascular plants. So basically, what does this lignin do? Basically, this lignin you will find in the cell wall. Okay, but in bryophytes, it lacks the lignin. Means lignin molecule is not there, and due to that, they cannot grow taller like the vascular plants. This lignin gives support, to hold a plant. They give the support, that they give the rigidity. But as bryophytes lack this lignin, they do not grow tall like the vascular plants, like the trees you see in our surroundings. They are tall because there is presence of lignin, but it lacks lignin, so they do not grow taller. So ecological importance, they are pioneers in colonizing bare or distributed soils and play a role in soil formation. So this bryophytes also helps in the, mainly their role is in the soil formation. Bryophytes also retain water and provides habitat for small organisms. They are also a habitat, means they also give shelter and food to the small organisms. Okay, this is all over the general characteristic. Okay. Next is cell structure of bryophytes. So this is very important part because general characteristic we have already done in our last class. Last means in the last chapter. So cell structure. So the cell structure of bryophytes shares common characteristic with other plant cells. Other plant cells means like pteridophytes and algae, but also they have some unique characteristic or unique adaptation to their non-vascular nature and life cycle. So let's go one by one. Number one is the cell wall. So the cell wall is made up of cellulose similar to plant cells. So very simple, cell wall is made up of cellulose. Function of this cell wall provides structural support and helps maintain the shape of the cell. So what does this cell wall do? They provide structural support and helps maintain the shape of the cell. They give shape to the cell. Unlike vascular plants, bryophyte cell wall lignin, which limits their ability to grow tall. So I have already told you that like the vascular plants, you here lignin is absent. So this is a like cellulose is present in the other plants also, in bryophytes also. But what is the difference? Difference is that here you will not find the lignin. So it limits the growth of the plant. The bryophytes are not taller because lignin is absent. Next is plasma membrane. So this is semi-permeable membrane that regulates the movement of substance in and out of the cell. So means they semi-permeable means they will not allow all the substances to go inside or to outside. Means they are semi-permeable. They are selective. Suppose they will allow calcium to go inside, but sometimes they will not allow iron to go inside. So that is called membrane selectivity. So plasma membrane has a membrane selectivity. They are semi-permeable. They will regulate whatever is needed inside or outside. So that is what plasma membrane do. Number 3 is cytoplasm. So the gel-like substance inside the cell that houses the organelles, and it is the site of most cellular processes. So basically all the membrane-bound organelles that are found in the gel-like substance, means in the matrix of the cytoplasm, you will find all the organelles, and it is the site of cellular process. So which is the site of cellular process? The answer is the cytoplasm. Next is nucleus. So it contains the genetic material, DNA of the cell. So nucleus is basically responsible for controlling cell growth, reproduction, and other essential process. And it contains the main thing, which is the DNA, the genetic material. So that sometimes plants are identical. Like the progeny is very much identical, means the baby is very much identical to the parent plant. Why? Because of the genetic material DNA. Okay, and this DNA is inside the nucleus, and it is responsible for controlling cell growth, reproduction, and other essential process. Next is chloroplast. So what this chloroplast do? So basically these are the main thing due to which these bryophytes or algae, these are the primary producers. They can produce their own food because this chloroplast has the chlorophyll. And this chlorophyll, we have already discussed chlorophyll in algae, and this chlorophyll molecule captures light energy for photosynthesis. Without chlorophyll, there will be no photosynthesis and there will be no production of food. So this chlorophyll is present inside the chloroplast. Next, plastids in bryophytes are responsible for starch storage and synthesis as in other plants. So what does plastids do? These plastids are also a part of the chloroplast. So basically these plastids are responsible for starch storage. Starch is a, like glucose kind of thing, which gives energy and synthesis in other plants. The number and arrangement of chloroplasts vary by species. For example, liverworts often have fewer chloroplasts per cell. Means how many chloroplasts will be there in a bryophyte, it is varied from species to species. Like suppose some has little amount of chloroplasts, but some plant you will find more number of chloroplasts. So it will vary from species to species. Next is vacuole. This is a large central vacuole is present in bryophyte cells, which maintain turgor pressure, store nutrients, and aids in waste removal. Basically, this vacuole is also useful for the storage. It stores the nutrients and maintains the turgor pressure and aids in waste removal. Like it helps for the removal of the waste. That is the role of the vacuole. Vacuole also help in the regulation of water content, critical for bryophytes given their moisture dependence. So basically, these vacuoles also help in the regulation of water content. I mean, how much water is needed in the plant, it is helped by the vacuole. Next is endoplasmic reticulum. That is also known as ER. In short form, endoplasmic reticulum is known as ER. So the rough ER is involved in the protein synthesis, while the smooth endoplasmic reticulum functions in lipid synthesis and detoxification. So basically, there are two types of endoplasmic reticulum. When we will study the cell biology, we will know there that the rough ER, it is responsible for protein synthesis. It will synthesize the protein, while the smooth ER, it will synthesize the lipid. Okay. So next, the Golgi apparatus. So this Golgi apparatus help in processing, packaging, and distributing proteins and lipids. Suppose endoplasmic reticulum has synthesized some protein. Okay. So now this Golgi apparatus, this will collect the protein and it will pack it and it will distribute the protein to the destination where it is needed. So this is the role of the Golgi. Next is peroxisomes, involved in the metabolism of hydrogen peroxide, a byproduct of cellular respiration. So basically, this help in the metabolism of hydrogen peroxide, and it is the byproduct of cellular respiration. So in physiology, we learn the cellular respiration and all. So these peroxisomes are the part of this. Next is plasmodesmata. So what is this plasmodesmata? So these bryophyte cells are connected by plasmodesmata, cytoplasmic channels that allow the movement of substances between adjacent cells, facilitating communication. So basically, this plasmodesmata helps in communication. Suppose there are many cells in bryophyte. Okay, so it will give a channel to the adjacent cells. So due to these adjacent cells, the movement of substances will take place. So this is the role of the plasmodesmata. Apart from this, there are some specialized cells in bryophytes. Okay, so what are these specialized cells? So number one is the rhizoid cells. Rhizoids are root-like structure mainly composed of elongated tubular cells. So rhizoids, I have already told, these are the root-like structure, but not roots. So this is composed of elongated tubular cells. So cells are elongated and tubular in structure. They help anchor the plant to the substrate but are not specialized for water or nutrient absorption. So their main role is to anchor the plant to the substrate. Okay. So these are the rhizoidal cells. Next is hydroids and leptoids. So in some mosses, some species of mosses, you will find hydroids. So hydroids are water conducting cells. Means they will help in dispersion of water. And leptoids are nutrient conducting cells. So this bryophytes generally they are non-vascular. Okay, so they don't have xylem and phloem, but they have these hydroids and leptoids. So these are not true vascular tissues but represent a primitive form of transport tissue. So basically here the evolution is taking place. In algae there is nothing like like water conducting cells or nutrient conducting cells, but in bryophytes you will not find the vascular tissue, but yes, you will find water conducting cells, which is hydroids, and the nutrient conducting cells, which is leptoids. So these are not true vascular tissue, but it is a very primitive form of transport tissue. Okay, so you remember the term hydroids and leptoids. You will not find these two terms everywhere. So hydroids transport water, while leptoids move nutrients within the plant body. Hydroid acts like the xylem, which transport the water, while leptoids acts like the phloem, which move nutrients within the plant body. This is hydroids and leptoids. Next is parenchyma cells. So photosynthetic parenchyma. These cells are abundant in bryophyte leaves and perform photosynthesis. So these photosynthetic parenchyma cells are very much abundant. You will find this very much in the leaves, and these perform the photosynthesis. Next is storage parenchyma. This cell stores nutrients and are present in both the gametophyte and sporophyte. So this storage parenchyma, you will find it in gametophyte also, sporophytic generation also, and it stores the nutrients. But the photosynthetic parenchyma, you will find this only in the sporophytic generation, in the leaves. Okay, and it will perform the photosynthesis. Next is sporophytic cells. So sporophyte structure. What is the structure of the sporophyte? Means what is the structure of the bryophytic plant? So the sporophyte, which grows out of the gametophyte after like this I have already mentioned that sporophyte is dependent in the gametophytic stage. So the sporophyte grows out of the gametophyte, is simple in structure, very much simple structure. Only three things are there. Number one is food. The food is responsible, anchor the sporophyte to the gametophyte. With the help of the food, the sporophyte is anchored to the gametophyte. Next is the seta. It is a stalk that supports the capsule. It is a stalk. Next is the capsule, which is the sporangium. So this capsule contains cells that undergo meiosis to produce spores. So basically the spores are present inside the capsules. Okay. And that undergoes meiosis. So food, seta, and capsule. Only three things in the structure of sporophyte of the bryophytes. Next is reproduction. So bryophytes have a unique reproductive cycle that involves alternation of generation, where both a haploid gametophyte and a diploid sporophyte phase occur. So here you will also find both haploid gametophytic generation and diploid sporophytic generation. So alternation of generation is present. Their reproduction include both sexual and asexual methods. So sexual reproduction is also there, and asexual reproduction is also there. Alternation of generation. So what is this? Haploid gametophyte, the dominant and photosynthetic phase of bryophytic life cycle. It produces gametes, sex cells. So in gametophytic generation, means who is haploid, 1N. So what happens there? Here, the bryophyte will produce the sex gametes, means the sperm and the egg. So sperm and egg is produced in the haploid gametophytic phase. After production of this, next phase is diploid sporophyte. Means dependent on the gametophyte, it produces spores through meiosis, giving rise to new gametophytes. So after the production of spores through meiosis, again new gametophyte is produced. So this is the alternation of generation. Okay, so let's learn more deeply. So number one, sexual reproduction. Bryophyte exhibit a true oogamous sexual reproductive system. So what is oogamous? We have learned the term oogamous, anisogamous, and isogamous. So oogamous is where there are two type of gametes. One is large non-motile eggs and the other is smaller motile sperm. Egg is non-motile, means it can't move, while the sperm is motile and it is smaller. It can move. So this is oogamous. Okay. So number one, gametophytic stage. The gametophyte generation is haploid N is the dominant phase. It is the main phase. It produces specialized reproductive organs. Antheridia, male reproductive organ that produce flagellated sperm. So this antheridia will produce flagellated sperms. Flagellated means the sperm has flagella. Okay, and due to this flagella, they are motile. They can move in the water and they can fertilize the egg. Next is the archegonia. It is the female reproductive organ that produce the eggs. Okay, so in archegonia, the eggs are produced. Both the antheridia and archegonia are multicellular structure. They have more than one cells, typically found at the tips of the gametophyte. At the tips of the gametophyte, it is found. After the formation of the gametes, next is fertilization. So which is water dependence. Bryophytes require a moist environment for sexual reproduction because the sperm are flagellated and must swim through water to reach the egg. So sperm, I have already mentioned that the sperms can move, they are motile and they swim in the water to reach the egg. So process is the sperm released from the antheridia. So once the antheridia is matured, the sperm will be released. It will swim towards the archegonia in response to chemical attractants, a process called chemotaxis. So there is a process named chemotaxis where the sperms get attracted to the female egg. So how will the sperm know that where is the egg? So due to this process chemotaxis, so release of some chemical attraction, chemical attractants, the sperm will know that there is the egg and they will swim towards the egg. Then the fertilization occurs when a sperm enters the archegonium and fuses with the egg. Next, the sperm will enter to the archegonium and it fuses with the egg, forming a diploid zygote, which is twice N, means diploid. So the process, here the main term is the chemotaxis. This is the process in where chemical attractants are releases by the archegonia and the antheridia will swim towards it. So this is chemotaxis. And then fertilization will take place and zygote twice N will be formed. Next, sporophytic stage. So gametophytic stage is over. Now we will learn the sporophytic stage. After fertilization, means zygote is formed. The diploid zygote remains within the archegonium and develops into the sporophyte. So it will remain in the female reproductive system and then it will develop to the sporophyte. The sporophyte grows attached to and dependent on the gametophyte for nutrients. So this sporophyte is dependent for the nutrients to the gametophytes. The gamete will, the archegonium will provide the nutrients to the sporophyte. So we have discussed food that is help the anchor the sporophyte to the gametophyte and absorb nutrients. Due to this food, the sporophyte can absorb nutrients from the gametophyte. Next is seta, that is a stalk that raises the sporangium capsule above the gametophyte. And the last one is the capsule, sporangium. The capsule is where meiosis occurs to produce haploid spores. As this sporophytic generation is twice N, means diploid. So in the capsule, meiosis will occur and again haploid spores will produced. In some species, there is a calyptra, a protective covering derived from the archegonium that covers the capsule. In not all species, but in some species, there is an exception where you will find calyptra. This is what is the calyptra? Role of the calyptra is a protective covering and it is derived from the archegonium that covers the capsule. So basically it gives protection to the capsule where the spores are formed. Next is spore dispersal. Inside the sporangium, diploid cells undergo meiosis, forming haploid spores. So meiosis will take place inside the capsule and again haploid spores are produced. So dispersal mechanism. The capsule releases spores into the environment, often using specialized structures like peristome teat. Okay, this is a very new term, peristome teat, that helps in gradual spore release. So in some mosses, you will find this peristome teeth. So this is a teeth-like structure, and this helps. What is the role of this peristome teeth? This helps in the dispersal of the spores to the environment. The peristome teeth will help the spores to disperse in the environment from the capsule in bryophytes. Once released, these spores germinate in favorable condition to form a protonema, an early filamentous stage from which the new gametophyte develops. So after it is dispersed, the spores are dispersed in the environment. Again, it will land to some favorable condition and germinate. Firstly, it will form a protonema stage, which is the early stage, and from this protonema, a new gametophyte will develop. So this is the life cycle, sexual life cycle of the bryophytes. Production of the gametes, fertilization will take place, then sporophyte will occur where the sporophyte is attached to the gametophyte. Then after that, the spores will produce in the capsule, and these spores will disperse in the environment by peristome teeth, and then it will reach in favorable condition and it will germinate. And first protonema will form, and from protonema again the gametophyte will form. So this is the whole life cycle, means sexual reproduction of the bryophytes. Next is asexual reproduction, which is very much easy and common like earlier. So bryophytes also have efficient asexual reproductive strategies to ensure the survival and spread of the species in favorable condition. The number one is fragmentation. So you know what is fragmentation? So part of the gametophyte breaks off and develop into new plants. So a part will break off and it will develop into new plants. So common in bryophytes, where any part of the plant can regenerate into a new gametophyte under suitable condition. So it is very common asexual procedure in the bryophytes. That is the fragmentation. A part will break off and it will develop into a new plant. Next is gamete. In liverworts and some mosses, many bryophytes reproduce asexually through specialized multicellular bodies called gamete. So these gametes are specialized multicellular bodies you will find in the bryophytes. So gamete cups in liverworts like Mercantia, gamete are produced inside gamete cups. So these gametes are formed inside gamete cups, which are located on the surface of the gametophyte. Means on the surface of the gametophyte, these gamete cups are located. When rain splashes into the gamete cups, the gamete are dispersed upon landing on a suitable location, grow into new gametophytes. So what happens what is the procedure? So when it will get some rain, the gamete will disperse in the environment, and when it will land on suitable location, it will again grow into new gametophytes. So this is a bit new concept of the gamete, which is formed inside the gamete cups, and when rain, it will splashes into the rain, it will disperse in the environment in a new location and grow into new gametophytes. Next is bulbils. In some mosses, certain mosses produce bulbils, small reproductive structures that can grow into new gametophytes. It is a very small reproductive structures. Next is tissue regeneration. Bryophytes possesses a remarkable ability to regenerate from fragments. Even a small piece of a leaf or stem can develop into a complete new plant. Suppose we human being. Suppose our one finger is cut. Okay. So from that finger, a new human being can't be produced. Okay. But in bryophytes or in some plants, what happened? Even a small piece of leaf or stem can develop into a complete new plant. This is called tissue regeneration. This is due to the totipotency. Means this is the capacity of the cells to regenerate into a new plant. So this is a very unique features of these green plants like bryophytes. And special features in reproduction. So the protonema stage, this is the special features in the reproduction. So I have already mentioned that after the spore germination, bryophytes form a filamentous or plate-like structure. Filamentous or plate-like structure called the protonema. This stage is the precursor to the mature gametophyte. Means the earlier stage of the mature gametophyte, which is the protonema. In mosses, it consists of thread-like filaments that resembles green algae, from which buds grow from the gametophyte. In mosses, like in some species of bryophytes, this protonema stage is like thread-like filaments, and it is very much similar to the green algae. So from this line, we can also understand that from this green algae, somewhere the bryophytes are linked. It also signifies the evolution, how evolution has take place. So this is also a proof of evolution. So this is the protonema stage, means the precursor of the mature gametophyte. Next is sporangium specialization. Mosses have elaborate sporangia that release spores gradually, ensuring more effective dispersal. So the sporangia is a bit specialized and that release spores gradually, ensuring more effective dispersals. Liverworts have elaters, elongated cells that help in spore dispersal by twisting as they dry, propelling the spores. So, liverworts, they have a structure known elaters. Okay, liverworts have a structure named elaters. So what these elaters do, these are elongated cells and they help in spore dispersal. Means once the spore is matured, they help to release the spores into the environment by twisting as they dry. Means suppose when the spores are matured, the structure will dry and it will twist. As a result, a force will create and the spores will release to the environment. So this is the role of elaters, which is present in liverworts. This is a specialized structure that helps in reproduction. So sexual reproduction involves water-dependent fertilization process where sperms swim to eggs, leading to the formation of a sporophyte that produce spores. While asexual reproduction occurs via fragmentation, gamete, bulbils, and regeneration, allowing bryophytes to spread quickly without the need for fertilization. Obviously, asexual reproduction is more faster and it spread quickly without the need for fertilization. So spores are the primary means of dispersal and the life cycle alternates between a dominant gametophyte and a dependent sporophyte. So sporophyte is dependent on the dominant gametophytic stage in bryophytes. The dual modes of reproduction allows bryophyte to adapt and thrive in various environments from moist forests to extreme habitats like deserts. So as there is a dual mode of reproduction, this helps the bryophyte to survive in harsh condition from moist forests to extreme habitats like also deserts. Okay, so this is the beneficial of the dual mode of reproduction. Next, we are done with the reproduction. So next is the classification. So bryophyte is basically classified into 3 classes. So number 1 is the Hepaticopsida, which is also known as liverworts. So sometimes you will find the term liverworts, which is the more common, or sometimes you will find the term Hepaticopsida. So you will understand that both are similar. Okay. So what are the characteristics of this Hepaticopsida? So there are specialized characteristics like gametophytic structure. Liverworts can be thalloid, flattened, and ribbon-like. They might be, means the structure of the liverworts might be flattened or ribbon-like. You have seen the ribbon, so it might be ribbon-like or leafy. It can also be leafy. In leafy liverworts, the leaves are arranged in 2 or 3 rows. If the structure of the liverworts is leafy, then the leaves will be arranged in 2 or 3 rows. And rhizoids are unicellular and serve mainly to anchor the plants, not for absorption. So these rhizoids are for the support of the plant and not for absorption. Some liverworts have air pores on the surface of the thallus, which are simple, unlike the stomata found in the other plants. So, here air pores are present. Later on, when you will see the most advanced form, you will find the term stomata. So basically, these stomata are the sites, for the exchange of gases and for photosynthesis. So here you will find air pores. Means some teeny, small spores on the surface of the liverworts. Means on the thallus of the liverworts, you will find air pores. Small spores. These spores are responsible for the exchange of gases. Okay, so these air pores are present on the surface of the liverworts. Next is sporophytes. The sporophyte is simple, consisting of food, etc., and a capsule. We have already discussed. Next is elaters. We have discussed it. Specialized spiral cells in the capsule that assist in spore dispersal by twisting and untwisting as they dry. So these elaters help in dispersal of the spores by twisting and untwisting when they are dry. So these elaters are present. Next is reproduction. These are the distinctive characteristic of the liverworts, the gametophytic and sporophytic. Next is reproduction. So sexual reproduction takes place. Liverworts produce antheridia, male structure that produce the sperm. This antheridia will produce the sperm, and archegonia, female structure that produce eggs on separate or the same thalli. Either on the same thallus or on the different thallus, this will produce. Fertilization is water dependent. It will require water as the sperm must swim to the egg, as the sperm is motile. Next is asexual reproduction. Liverworts reproduce asexually through fragmentation and by producing gamete, multicellular reproductive bodies in specialized structure called gamecops. So in asexual, you will find fragmentation where a small part will break off and give rise to a new plant, or producing gamete, which is found inside the gamecops. Next, alternation of generations. So there is dominant phase, the end phase, the photosynthetic thallus or leafy structure is the dominant phase where the gametes, male gametes and female gametes will produce. Next is the dependent sporophyte, that is the twice end. The sporophyte is short-lived and remained attached to the gametophyte. The sporophyte is the short-lived, means it is very much short. The dominant gametophytic stage is more long-lived stage, but the sporophyte is for few time, very less time, and it is attached to the gametophyte for nutrition dependent. For nutrition purpose, it is attached to the gametophyte, means it is dependent. Without the gametophytic stage, sporophytic stage can't survive. It is totally dependent on the gametophytic stage. I am talking about the liverworts, which is the Hepaticopsida, class Hepaticopsida. So example is Marsantia, a well-known thalloid liverwort with prominent gamecops. So this is the example, a genus of the Hepaticopsida with prominent gamecops. And also Richia, thalloid liverworts with simple structure. So basically, in liverworts, like in Hepaticopsida class, this Marsantia and Richia is very much famous and common genus. So the number 2 class is the Anthoceropsida, which is commonly known as hornworts. So don't confuse with the liverworts and hornworts. So this is second class is known as hornworts. So the main characteristic is the gametophytic structure. Hornworts have a thalloid gametophyte, which is flat, lobed and lacked distinct lips. So here you will not find distinct lips, and it is thalloid in structure. Rhizoids are similar to the liverworts. The rhizoids are unicellular. It is unicellular in structure. A key feature of hornworts is the presence of stomata on the sporophyte, which allows gas exchange, a trait shared with vascular plants. Okay, so here you see how evolution is taking place. In the earlier class, means in the class Hepaticopsida, means the liverworts, you have seen that there is present of air pores, which is similar to stomata, but a very primitive form. But here there is a difference. In the hornworts, you will find the presence of stomata, means how the evolution takes place from the air pores to the stomata. So basically, the stomata are a trait of the vascular plants. But here also in bryophyte, in the hornworts, you will see the stomata. So now the evolution is taking place, means to the advanced level. So you will find stomata. So next is sporophyte. The sporophyte is elongated and horn-like. And that's why it is known as hornworts, because the structure of the sporophyte is horn-like. It grows continuously from its base, even from the upper part has released spore. Means it grows continuously. Means there is no full stop in the growth. From its base, even after the upper part has released spore. After the spore is also released, it will grow. Unlike other bryophytes, the sporophyte can perform limited photosynthesis, making it semi-independent from the gametophyte. So basically, this is semi-independent, not fully independent, because it can somewhat produce somewhat produce, means perform the photosynthesis. It can produce their own food, so it is semi-independent but not fully independent. They are somehow dependent to the gametophytic stage. The sporangium splits lengthwise to release the spores. So here the sporangium will split lengthwise to release the spores. Next is reproduction. So sexual, here Antheridium and Archegony are embedded in the gametophytic stylus. Then the fertilization is water dependent with fecundated sperm swimming to the egg for fertilization. Exactly same to the liverworts, no difference. Next is asexual. In asexual, reproduction occurs primarily through fragmentation of the thallus. So here fragmentation will take place. Okay, so fragmentation will take place, means small part will cut off and it will give rise to new plant. So this is the asexual reproduction. Next is alternation of generation. So dominant gametophytic stage, that is the end. The thalloids photosynthetic gametophyte is the dominant generation, means it have more long time. And next is semi-independent sporophyte twice end. So here you will see a difference in the earlier class, that is the hepatic opsida, means the liverworts. I have mentioned dependent sporophyte, but here semi-independent, because it is photosynthesis is there, but this is limited. Not totally, means it can produce their own food. It is limited. It is also dependent to the dominant gametophyte, but it is semi-independent, not fully. That's why the term semi-independent is mentioned here. This is the difference, key difference. The sporophyte is horn-shaped, photosynthetic, and grows from the gametophyte, but still relies on it for nutrition. Means it can produce their food, but still it relies on the nutritions to the gametophyte. Means the gametophyte provide the nutrients. That's why it is known as semi-independent sporophyte. So the common example of this hornworts is the Anthocerus, a common hornwort with a simple thallus and horn-like sporophytes. Anthocerus. Okay, and here there is no elaters, means special spore dispersal mechanism, no elaters. But earlier, in the earlier thing, we have seen that there is presence of elaters. But here you will not find elaters. So this is the second class. So the third class and the last class is the Bryopsida, which is the Moses, which is the most advanced class of the bryophytes. So what are the distinguishing characteristics? Number one is gametophyte structure. So mosses have a leafy gametophyte with small simple leaves arranged spirally around a central axis or stem. So now it is advanced. Here you will see the leafy gametophyte, and also you will see simple leaves arranged spirally around a central axis or stem. Next is rhizoids, which are multicellular and thread-like. But in the earlier class, that is the hornworts, you have seen that the rhizoids are unicellular. So these are very small, differences. Here you will see multicellular. In the earlier, you have seen the unicellular rhizoids. So these are the small differences. So rhizoids are multicellular and thread-like, providing anchorage but not conducting water. It will give only support, anchoring support, but not conducting water. Mosses often have specialized protonema So here you will see the protonema a filamentous structure from which the leafy gametophyte develop. So from this protonema a new gametophyte will develop. The next is sporophyte. The sporophyte is well developed, consisting of a foot, seta, and capsule. So the sporophyte is very much developed because this is now a advanced form of the bryophyte. After this, we will see the next phylum, that is the pteridophyte. So this is the last class of the bryophyte. So it is most advanced. So sporophyte have foot, seta, and capsule. The capsule sporangium is often covered by a calyptra, a cap derived from the gametophytic tissue. So here you will see the calyptra. So what is this calyptra? So this is a protective structure that protects the sporangium, means the capsule. And inside the capsule, spores are produced. So the calyptra is a protective tissue that surrounds the capsule. And it also contains specialized peristome teeth that help release spores gradually. So what these peristome teeth do? They help to release spores gradually. In the earlier, Hepaticopsida liverworts, we have find there is a presence of elaters, which helps in dispersal of spores. But here is the presence of peristome teeth that help release spores gradually. So these are the unique features of the classes. Somewhere there is elaters like Hepaticopsida, you will find elaters, but here you will see the peristome teeth. So these are the main, like unique features of the classes. So reproduction. So mosses produce Antheridia and Archegonia on the gametophytes. So it will produce Antheridia and Archegonia on the gametophyte. Water is required for sperm to swim to the egg where fertilization occurs inside the Archegonium. So what happens? The male Antheridia will produce the sperm, and the female Archegonia will produce the non-motile large egg. So after the production of these motile sperms, this will swim in the water. It will get some, signals from the egg, and then it will fertilize the egg. And here the zygote will produce. Fertilization will take place, and zygote twice and will produce. This is the procedure of the sexual reproduction. Next is asexual. Mosses reproduce asexually through fragmentation, and in some species by producing gamete, although less common than in liverworts. So basically this gamete production, you will find more in liverworts, more common in Hepaticopsida. But in Bryopsida, it is present, but not in every genus. It is very less common. Mostly in asexually, it will reproduce through fragmentation. A small part will break off and it will give rise to a new plant. Next is alternation of generation. So dominant gametophytic stage and the leafy photosynthetic gametophyte is the dominant stage, means where the in the gametophytic stage there will be the production of the male gamete and female gamete. This is the dominant stage. Next, the dependent sporophyte. Here sporophytic is totally dependent to the gametophytic generation for the nutrients and remain attached to the archegonia. Okay, and this is a short-lived stage, the dependent sporophytic. So example is sphagnum, that is peat moss, important in box ecosystem, and polytrichum. This is hair-capped moss, known for its tall sporophyte and robust structure. These are the common genus, sphagnum and polytrichum. So this is all about the bryophytes, classifications, distinguished characteristic and all. So now I have collected some ecological roles of very much common bryophytes, which is like, like in exams, sometimes question is asked on the common bryophytes. So I have collected the common genus names, which is used widely and have commercial use. So number one is sphagnum, that is the peat moss, also common name is peat moss. So these are the species of the sphagnum. So sphagnum is the genus and these are the species. So Sphagnum palustre, Sphagnum fuscum, Sphagnum papillosum, and Sphagnum megalnicum, etc. So there are many species. So this is the sphagnum is the main genus. So you remember the term sphagnum. That is also known as peat moss. So they have very much commercial use in horticulture. Means it is widely used in soil conditioner and growing medium because its ability to retain water. So this sphagnum can retain water. It can hold water. It's often mixed with potting soil for plants that require high moisture, such as orchids. So these sphagnums are often mixed with some potting soils for plants which requires high moisture, like orchids. They require high moisture. So these sphagnums help them to retain the water. Next is peat production. Peat derived from particularly decomposed sphagnum is harvested for use in horticulture as a soil amendment and in biofuel production. So this is also used in biofuel production. That is, next is wound dressing. Historically, sphagnum moss was used for dressing wounds due to the absorbency and antiseptic properties during World War I. So this is a very important thing, that this sphagnum species, this genus is so much important. It is historically important because this is used in wounding. Some people get injured And when people are injured, these are used in the wounds, and it is used as antiseptic during the World War One. So this has historical importance. Next is water filtration. Peat mosses is used in some regions for water purification and filtering out contaminants. So this is also used as water purification and filtering out the contaminants. So contaminants are removed. Like that, Mercantia, which is Hepaticopsida. So these are the species: Mercantia polymorpha, Mercantia inflexa, and Mercantia pelagia. So commercial use, it is used in medicine, like liverworts extract specially from Mercantia polymorpha have been used in traditional medicine to treat liver ailments, respiratory issue, and wounds due to their anti-inflammatory and antimicrobial properties. So this Mercantia polymorpha has been used traditionally in medicines. Okay, next is they have also horticulture use in scientific research related to plant development and genetics. Next is Polytrichum, haircap moss. So common species are Polytrichum communion, Polytrichum juniperinium. So commercial use is bedding material, traditionally used in bedding material by indigenous people due to its softness and moisture retention. Next is it will also help in erosion control. Like it can give restoration of the soil, and it will help in managing the soil erosion. Next is roofing and insulation. In northern Europe, Polytrichum and other mosses were used in the construction of turf roofs for insulation. Means roofs are made for insulation. Means not so that it is not like it can conduct electricity. Number 4 is Richia, which is a liverwort. So common species is Richia fruitans, commercially is used as aquarium industries. So these are like widely used in aquarium as a floating plant. It provides habitat for fish and aquatic organism and helps in oxygenating the water. So they give habitat for fish and other aquatic organism. They also help in water filtration. It also acts as biofilter in aquariums, removing excess nutrients and removing water quality. So this is Richia. Next is Funaria, which is also known as Hort moss. So species is Funaria hygrometrica, commercial use in education and research. So this is used as a model organism in botanical studies and education due to its fast growth and easily observable life cycle. So this Funaria can be easily observed. The life cycle can be easily observed, and it is very fast growing. That's why it is used as model organism in botanical studies, and it is also bioindicators of pollution in environments. Suppose in a environment, suppose there are funaria, and suppose you see that the funarias are dying because there is a rise in pollution. So this is also used as bioindicators. Number 6 is Anthocerus. So the Anthocerus are the species is Anthocerus punctatus. So commercial uses biological studies. So these hornworts are widely used in research to study plant physiology, particularly because of their nitrogen fixing capabilities through symbiotic relationship with cyanobacteria. So this Anthocerus, they made a symbiotic relationship with the cyanobacteria, and they are widely used in the research studies for study the physiology of the plant and for the nitrogen fixing capacity. So the cyanobacteria basically they help in nitrogen fixation. Means there are abundant nitrogen, but this has to be fixed in the environment. The plant can't absorb the complex nitrogen, so it is fixed by some bacteria, and this Anthocerus of bryophytes helped as it it makes symbiotic relationship with the cyanobacteria. So these are the some common genuses of the bryophytes which are commercially used. This is a very huge ecological importance. So basically there are there are more number of genuses, but all are not important. These are the very common and famous ones, so I have collected these things. Okay, so this is all about the bryophytes. So we are done with the bryophytes. So what we have discussed? We have discussed the general characteristic, the cell structure, detailed reproduction, both sexual and asexual, alternation of generations, then classes, the 3 classes with their distinct characteristic, reproduction and alternation of generation with examples, and the ecological importance of some common genuses. So we are done with the bryophytes. Okay, so this is all about the bryophytes. In the next class, we will continue with the next phylum, that is the pteridophytes, which is the next one. Hello, ma'am.
Yes, ma'am.
Yes, ma'am. Bryophyte is the class is done for today.
Okay, okay, ma'am. Yes, ma'am. Thank you, ma'am. Again, we have class tomorrow morning, that is from 5 to 7. Right? Don't miss the morning classes. You have chemistry and physics.
Okay, ma'am.
Okay?
Okay,
Yeah.
ma'am.
Okay. So again, we'll meet tomorrow morning, that is from 5 to 7.